I think we can get started. Hi, everyone. Good morning, and welcome to the Single Cell Omics Germany seminar series today. We are just waiting for some people to join still, but it's really a pleasure today to be able to hear a lecture from Christian Schmidl. Christian is coming from the Leibniz Institute for Immunotherapy from Regensburg, and he's a group leader there who started his lab just 5 years ago in 2017, and his research focus is to explore the epigenetic programs that are regulating immune cells in oncology and the tumor microenvironment. So before we get started on the lecture, I first wanted to do a little housekeeping. So first, next week, or pardon me, the next lecture series will be held from Heiko Leichert, who is scheduled for May 20th, later this month. And he will be talking about the beta cell development and regeneration. And so he'll be giving that lecture in 2 weeks. So I hope that you can join us for this. And also, we're very happy to announce the first next in-person workshop, which is scheduled for September. This will be in Munich this year. And of course, we're all very excited to have an opportunity to meet in person again and to bring the community together. So this is really just save the date in advance of this for September 12th and 13th in the fall. And we already have 4 fantastic speakers confirmed for this. And many more will be added to the program in the future. So definitely keep an eye out for the announcement, and the registration will be opening soon. So we are really looking forward to that. And so now I just wanted to give a little information about how the lecture will proceed today. So as normal, we have a 30-minute presentation that's followed by a 15-minute discussion period. And we really hope for an active discussion and question period there. So please do use the raise hand feature if you would like to ask anything or pop in with some discussion. And also, you can also, of course, write your questions in the chat, and I would be happy to read them out for you if you like. And also, today's seminar series will be recorded as normal. So that's out of the way now. So once again, I will take a moment to welcome Christian for today's lecture. We're really excited to hear about some of your recent work on using single cell genomics, of course, to dissect single cell or cell state enhancers and the landscapes of human T cells that are infiltrating tumors. So I will open the floor up now to Christian. Thank you.
Thank you very much, Ashley, for this nice introduction. And thank you for giving me the opportunity to speak today. I'm going to share my screen now. Okay. And thank you, of course, for tuning in today. I guess you can see my screen and hear my voice.
Yep, looks good.
Welcome, everybody. I would like to present some of our recent work that we started roughly 4 years ago in my junior group, where we're looking at the chromatin landscape of T cells that infiltrate tumors. So as a short, sorry, slide is not moving. Oh, now it is. As a short overview of my talk today, I would like to introduce the concept of T cell dysfunction or T cell exhaustion. And in the second and largest part, I would like to present some data on the approaches, how we try to tackle this topic by single cell chromatin profiling of human tumor-infiltrating lymphocytes. In the third part, I would like to show you how we try to use this information to get a better understanding how genes are regulated in T cells. And in very short last part, I would like to convince you that this information bears useful information that we can use to maybe improve adaptive cell therapy in the future. So T-cell dysfunction, T-cell exhaustion, why is this important? You are aware, of course, that the immune system, especially T cells, they can recognize tumor cells and lyse them. And this happens by the tumor cells having DNA mutations, producing aberrant proteins that can then be recognized as foreign by the T cell receptor of tumor-specific T cells. The T cells become activated. They also need co-stimulation and cytokines. And upon full activation, they expand, produce effector molecules that can then kill the tumor cell. But it has been shown that this process is not always successful because tumors and T cells often coexist. And this is in part lying in the expression of inhibitory receptors that engage on the T cells, like PD-ligand 1 engaging PD-1 on the T cell, that is interfering with the T cell activation, putting a break into the T cell and preventing the T cell then from finally killing the tumor cell. And the discovery of those pathways that involve, for example, PD-1 and CTLA-4 were honored by a Nobel Prize 2018 for James P. Ellison and Tetsuko Honjo. And these discoveries, they have led to the development of medication, for example, blocking antibodies that really interfere with those pathways, that help them to take out the brake of the T cells, revive them, reactivate them, and make them successfully kill the tumor cells. And this gave opportunity to really help in several cancers where nothing could be done previously. So metastatic melanoma was basically a death sentence, but with the immunotherapy, using these blocking antibodies, you have, at least for some patients, a long-lasting improve in survival. And the same is true, for example, for lung cancer, where versus chemotherapy, the blocking antibody against these inhibitory pathways really significantly improved the survival. And you're right when you say still many of those patients do not respond. And one part of the reason for that has been investigated for a long time now. And people came to the conclusion that when the T cells, they become dysfunctional or exhausted, they are also changing drastically their epigenetic landscape, including the chromatin landscape and DNA methylation landscape. And this has led to the suspicion that maybe this fixes the T cells in such a dysfunctional state that they cannot be revived by immunotherapy. There is some really nice data already published on this topic, for example, from a mouse model from Andreas Schittinger's lab, where they have an autochthonous tumor model where liver cancer develops. They profile T cells along this tumor development. So they take the tumor-specific T cells out of the tumors, and then you see that PD-1 in the course of tumor-infiltrating T cell development and persistence is upregulated. And we see a complete loss of cytokine production. So when you here look at interferon and TNF double positive cells, which is a measure for T cell functionality, you see that in an infection model of effector cells, they almost to 80% or more can produce these cytokines. But in the tumor, already after 5 days, you completely lose the cytokine production. And this dysfunction is accompanied by chromatin remodeling. So you have 2 stages where the chromatin drastically changes, at least in this murine model. There was also a recent paper that did already single cell profiling of T cells from tumors. This is patients with basal cell carcinoma, pre and post immunotherapy. And based on the data, the authors, they drew conclusions about a very rudimentary development of those T cells, of CD8 T cells that either go into the effector, memory, or exhausted state. However, the situation is more complex than that, as we know now, which is nicely summarized by this picture from a review from Dietmar Zien. So if the T cell not goes into the effector or normal memory route, but goes down the road to exhaustion, it is now kind of accepted knowledge that there are exhausted progenitor cells that play a really important role in the response to therapy and also in clinical outcomes. So the terminal exhausted cells, the final dysfunctional state, is pretty different from this progenitor exhausted T cells. So the progenitor exhausted T cells can still renew themselves. They express lower levels of PD-1, but this is important, they express CXCR5 and the transcription factor TCF7 or TCF1. So TCF7 encodes for TCF1. While the terminally exhausted cells, they have really high levels of PD-1, they lose CXCR5 and TCF7 expression, and importantly, they upregulate TOX, which is also a transcription factor that is important for T-cell exhaustion. And also in the human system, a high prevalence of TCF1, so marker of the progenitor exhausted cells, is associated with a better survival probability. And it is now kind of accepted that high TCF1 or TCF7 expression in those T cells is characterizing T cells that can respond to immunotherapy, but also it has a prediction for the patient outcome in these settings. And coming back again to TOX, high levels of TOX, which is a marker for terminally exhausted cells, is associated with a lower survival probability in other cancer entities. So the heterogeneity of the exhausted T cells comprises several treatment-relevant states, but in the human system, this information is not as complete as we want it to be. And that's why we profiled the single-cell chromatin landscape of human tumor-infiltrating T cells to a detailed extent. So this work was carried out by Dania Riegel, which is my first PhD student, and she's finishing now. And Dania took from several clinical collaborators where we got cancer specimen samples from head and neck cancer, liver cancer, renal cell cancer, and also control CD8 T cells isolated from the blood. And she then isolated from single cell suspension immune cells. And just to give you a functional assessment of the data by flow cytometry, you can appreciate that the CD8 T cells taken from healthy blood, if you pre-activate them, they upregulate, on this axis is PD-1, they upregulate PD-1, and they can secrete cytokines. So this is now interferon gamma, so they produce a lot of interferon gamma. When you go to the other end of the functional spectrum, when you look, for example, for the hepatocellular carcinoma tumor-infiltrating T cells in dark green, you see that those T cells are high in PD-1 and they are low in cytokine production. So most of these cells are cytokine negative. When you take T cells from the same biopsy, but not from the tumor, but from the healthy tissue that is also taken in tiny pieces as part of the normal biopsy, you can see that these cells, they also can in part upregulate PD-1 when they are activated, and they are all basically interferon gamma positive, meaning that they're functional. And when you do this for all the samples that we then finally put in the chromatin analysis, you see that there is a negative correlation between PD-1 expression and the cytokine production. So we have here, for example, this sample has very high protein expression of PD-1, but very low double positive interferon TNF positive cells. And you see that this correlation for the tumor sample is an inverse one, while for the T cells from the blood, this is not the case. And you also, I hope, can see that this is not a black and white situation, what we have here, but we have like a gradient of functional T cell states, meaning that the data set then we finally analyze with RNA-seq is covering the functional breadth of the T cell states in a good way. So we then, from those samples, isolated, sorted CD8 PD-1 positive and CD8 positive PD-1 negative T cells and subjected them to an optimized plate-based single-cell RNA-seq assay to profile the open chromatin landscape. And at this time point, the 10x kit was not available yet. So we did this all by plate-based assays, and 20,000 PCRs later, we came up with this clustering. So these are now all CD8 T cells from healthy donors, but also from the tumor. And in total, we find 11 clusters here. And when we look at the entities, we see that some clusters like this one here, they comprise cells from all of the entities and also from the healthy donors. In addition, this cluster here looks the same in terms of distribution, while we have other clusters that are really dominated by a certain entity. And we analyzed all this data, or most of this data, with Archer, which is a single-cell RNA-seq analysis software from the Greenleaf Lab, which I think is really nice. And it uses a model to predict from the open chromatin regions gene expression. And there, when we look at marker genes, we find many things that make sense. For example, ZBTB16 is a major transcription factor for mucosa-associated invariant T cells. So cluster 7 comprises mate cells, mucosa-associated invariant T cells that are not classical CD8 T cells and not of so much interest here. We have LEF, which is basically a marker for naive T cells in cluster 6. We have CX3CO1, which is a marker described for cytotoxic T cells in cluster 9, where we have all the entities represented. Then we have 2 clusters that show strong activity of heat shock factors. This would be cluster 8 and cluster 3 here. And then we have the expression of exhausted progenitor markers CXCR5 and TCF7 in the non-naive cells in the tumor in cluster 10 and 11. And then when we look at the cluster 1 to 4, we see a strong activity of T cell exhaustion markers like HAF-CR2, which is encoding TIM3, and PD1, which is a classical exhaustion marker, and also TOX, which I introduced in the introduction. And just to give you a visual impression of the data, when we sum up the open chromatin data from those different clusters, you see that in the clusters 1 to 4, which I here labeled as dysfunctional or exhausted, you see that at the TOX locus, which is really a huge gene, you see a strong increase in chromatin accessibility in comparison to other clusters, especially the naive, but also other memory or cytotoxic T cells. And when we compare the gene activity to public gene expression databases that contain signatures of certain cell states, we can more or less validate our findings. So we see in cluster 6, we have gene activity of genes that have been described in naive T cells. We have cytotoxic T cells in cluster 9, as I said. And then we have all dysfunctional or exhausted cells signatures enriched in clusters 1 to 4. Interestingly, these clusters also contain signatures of genes from T cells that do not respond to PD1 blockade. So which transcription factors now regulate those cell states? ATAC-seq is a nice tool to study this. So for this, we cluster, we call peaks for each cluster, and then we use ChromVar to look for transcription factor motif deviations. So basically, we look for a transcription factor motif in all open chromatin regions in each single cell and see at especially this transcription factor motif is if in one single cell we have more or less accessibility at all those putative binding sites. And for that, we can then get an impression how active this motif is in a single cell. And when we plot the TF motif deviations, we see also things that make sense, but also new interesting things. For example, ROGAMA-T is a transcription factor also active in mite cells, and this is really highly active here in cluster 7. We have TCF motifs in cluster 6 for the naive T cells, and then we have NF-kappa B and NUR77, which is encoding NR4A1, T cell receptor activation dependent transcription factor that is highly active in clusters 1 to 4. And NUR77 has been described to be a driver of T cell exhaustion, so this fits with the literature. What is kind of interesting is that KLF is lost in the exhausted T cells, which has not been described yet in detail. And we also observe that heat shock factors, the transcription factors that are activated upon a heat shock response, are strongly active in those 2 clusters here. And then we can also look at transcription factor footprints. So when you're familiar with ATAC-seq, ATAC-seq uses the transposase to insert sequencing adapters basically in open chromatin regions that are nucleosome free. And then we get these nice peak-shaped browser tracks that we can visually inspect. But when you look at the single base pair resolution of this data, you will also see that within these peaks, you have at the points where transcription factors bind to their motif. If this protein really binds there, it protects also the DNA from being segmented by the transposase. So it protects from ATAC sequencing. And this gives you then an enrichment of the signal in this peak, but a dip of the signal at the exact transcription factor binding motif, which suggests indirectly that the protein is really binding there. And we can then confirm the binding maybe of TCF, of START, NF-kappa B, NUR77 in some specific clusters. And what I think is pretty eye-catching is that these 2 clusters really show a strong footprint for the heat shock response proteins. And this is interesting because tumors are often inflamed, and there was a recent paper that showed that heat or fever, so heat for some time, is making the T cells better in tumor control. So this is maybe worth investigating further. So what we did also observe is that the UMAP segregates basically the exhausted T cells on UMAP dimension 1. And we thought this could be really interesting because basically the exhausted T cells that are in the end lysing the tumor cells even if they arise from the progenitors. If they have different flavors, there could be some function in it. We are not there yet. We are just describing at the moment because our data set is not big enough to make final conclusions, but we observe still several thousands of differentially active open chromatin peaks between those cluster or entity-dominated clusters. Then we have a look at enriched transcription factor motifs. We see that, for example, the T cells that infiltrate the skin, they show nuclear receptor or octamer transcription factor motifs enriched, while the liver shows a dominance of T-box and RUNX motifs, while we find NF-kappa B, for example, in the renal cell carcinoma. And this could be really interesting from a therapy and patient perspective, as the further we go towards exhaustion, the more the tissue seems to have an impact on the chromatin profiles of the tumor-infiltrating T cells. But initially, we actually wanted to see if we can find a unifying signature that is leading to T cell dysfunction that is valid over several entities. So we did remove the mate cells, which are not of interest here, we removed the PBMCs, and we tried to correct for the tissue effect, and then we ended up with this kind of UMAP and clustering. When we inspect marker genes, we're pretty sure that the cluster 1 and 2, they contain exhausted T cells, because we have here expression of PD1, TIM3, TOX, CTLA4, and PD1, the markers that I already introduced. We have cluster 5 with the heat shock signature, so this would be this cluster here. Cluster 6 that now contains the short-lived effector cells with all the cytotoxic granulae that can then kill the cancer cells in addition, but that are very short-lived and maybe not tumor-specific. And then we have cluster 3 and 4, which carry more memory and T-X progenitor markers. So cluster 3, for example, we suspect to be the precursor exhausted cells because they have the highest level of TCF7 and CXCR5. Further evidence that cluster 1 contains the terminally exhausted cells is the protein information we have from the cell sorting. Remember, we FACS sorted the cells in PD1 positive negative, and virtually all of the cells in those clusters are PD1 positive. And from the public data set, we also integrated the basal cell carcinoma single cell ataxia. We see that cluster 1 is basically expanding post-immunotherapy, which is in line with the data that has been shown that the precursor exhausted cells, they have a proliferative burst upon checkpoint blockade, and then terminally exhausted cells, they expand, and they, although they are impaired in their mass and still with the remaining cytotoxicity, they get rid of the tumor. So this is also in line with what is known. And we have also control cells from the tissue, when you remember the FACS plots from the beginning. So when you take T cells that were from control tissue of the kidney and the liver, they hardly populate cluster 1, and is mainly populated by the tumor. So we think, based on this evidence, that we have kind of a trajectory that goes from the precursor exhausted T cells from cluster 3 towards cluster 1. And then when you compute this dysfunction trajectory, and we follow genes along the pseudotime axis, we see that TIM3 as an exhaustion marker goes up along this trajectory, while TCF7, which is prevalent in precursor exhausted cells but lost in the terminal exhausted cells, is also going down as expected. So these are only 2 examples of 2 genes. We can do this, of course, along this trajectory for all the peaks. Then look at enriched transcription factor motifs within those significantly changing peaks. And we can also do this on a gene-based level to identify known, like TCF7, for example, or TOX, but also many new genes that are putatively involved in the process when the T cell from the precursor state to the terminally exhausted cells lose their function. So I hope in a first summary, could convince you that the single cell chromatin profiling is a really good tool to discover the heterogeneous cell states that we observe in the tumor, that we observe a potential impact of the tissue on the residing immune cell. And this has been shown for other immune cell types previously. For example, Ido Amit had a very nice paper on macrophages or monocytes, I think, that were transplanted from one tissue to the other, and then they acquired the phenotype of the resident cells. And also with Markus Feurer, we had a recent work on regulatory T cells that showed that chromatin accessibility and DNA methylation of regulatory T cells in the tissue is also strongly impacted by the target tissue where they live in. And in the end, I think I hope it made sense that we could identify this core pathway to terminal dysfunction that is valid over several cancer entities. So what we're going to do with this information, we now profiled the open chromatin landscape of the T cells, and we found many, many regions that were different between different cell states. And what we observe is that many groups, they do similar things, be it on the bulk or single cell level. So there are many nice papers that showed with really sophisticated models from T cells isolated from mouse models, human cohorts, that many regions change between T cell states, and exhausted T cells are regarded as a kind of a unique lineage that is different from functional memory cells or effector cells. And we do the same. For example, again, here, the TIM3 locus, we identify these exhaustion-specific regions that are putative enhancers for TIM3. But in the end, most of the studies, they fall short in really understanding what is happening. So we identify thousands of regions, but in the end, we have no clue what they are really doing. So what are the target genes of these putative enhancers and what is their impact on expression is what is basically for those thousands of regions that are discovered in many of those studies is not really known. And that single enhancers are important, you are probably sure, you are convinced that they are, but just to give you two examples, one from CD8 T cells, there was a paper from Jeremy Boss' lab that deleted an enhancer in mice upstream of PD1, which is encoded by PDCD1. And when they delete this 500 base pair regions, this completely prevented the T cells from being activated in vitro with the T cell receptor stimulation. But in vivo, the cells still could be activated, and they upregulated PD1 at a different kinetics than the non-enhancer deleted cells, which led in the end to a better memory T cell formation and a better tumor control. So this just by deleting one 500 base pair element of one gene in a T cell. And there is also a nice paper from Alex Marston's lab. They did enhance a screen of the IL-2RA gene, which is encoding CD25. This is a IL-2 high affinity receptor part, which is important for T cells to survive and expand. And they showed by deleting one of the enhancers that they found really changed the phenotype of the CD4 T cell compartment in mice when the mice were vaccinated in vivo. So this is just two examples of two single enhancers that change the immune response by simply taking out one non-coding region. And to work towards a better understanding what the enhancers are doing and how they control gene expression, this is the project of Elena Romero Fernandez, my second PhD student, who's been taking on this project. So the first step that we want to do is to link enhancers to promoters in silico. And for that, we integrated single-cell data that we had with matching public single-cell RNA-seq data from CD8 T cells from the same entities that we've been analyzing. And when we do this, Archer via Seurat is, I think, providing this option where by the integration of RNA and ATAC on a single-cell level, the program is building pseudo multi-omic cells where you have then, based on the alignment of the gene activity, chromatin-based and the RNA expression, it builds single cells that have both the chromatin and RNA information. And with that, you can basically build calculate correlations of region is open, so enhance is open, and gene is expressed, or gene is closed. Enhance is closed, gene is not expressed. And then you can visualize those interactions or correlations by these loops, which are called peak to gene links. And for example, here at EOMS, transcription factor, you can see that the output predicts maybe interactions of those downstream enhancers. And this is not a random assignment because these peaks that are close to the other side to EOMS, they are not predicted to have an impact on EOMS expression. So if those predicted enhancer promoter interactions in the end are true, we don't know. But of course, we try to validate this data based on publicly available other data sets. When we look at this IL-2RA enhancer paper, where I introduced one enhancer in the previous slide, this paper conducted or contains a CRISPR screen that investigated enhancers at the IL-2RA locus, which is visualized here. So the bigger the blue signal here, the higher is the activation score of the CRISPR activation screen, meaning these are kind of functional enhancers, which are highlighted in blue. And the peak to gene links that we calculate basically hit all of these enhancers that were described, and they all loop to the IL-2RA gene and not to the gene that is also very close to these open chromatin regions that is upstream of IL-2RA. So on the single gene level, in this case, the predictions make sense. All in all, we discover almost 50,000 of those peak to gene links by integrating this data. And then we try to kind of overlap our peak to gene links with physical 3D chromatin interaction data that was published. So this is an old paper that used promoter capture Hi-C to physically map interaction of gene promoters with enhancers in different immune cell types. And we see for CD8 T cells a specific enrichment of observed interactions that match the peak to gene links compared to random. So also on the genome-wide level, this seems at least to make sense in some degree. But of course, we need to validate some of those enhancers to be sure that the predictions are okay. But first of all, we try to use this information to get an idea about which of the genes are, or which of the enhancers are important in controlling a cell type specific or cell state specific gene expression. And for that, we use those peak to gene links that we calculated. Then we filtered out all the enhancers or the open chromatin regions that were not specific to a certain cluster. So we just used the cluster specific ones. And then we did a simple ranking of the genes based on the number of cell type specific enhancer promoter interactions. And when we then go back to the core clusters, and when we look, for example, at the remaining exhausted T cells in cluster 1, the result was actually much better than I anticipated. So the most highly ranked gene here is TOX. And we also find all the other genes that are involved and have been described to be really essential for T cell exhaustion, like CTLA4, TOX2, BATF, TIM3, and PD1. They are all among the top ranked candidate genes when we do this prioritization based on the enhancer promoter interactions. When we look at the precursor exhausted T cells, we find our candidates CXCR5 and TCF7 are among the top ranked one. And when we look for the short-lived effector cells, we find described candidates from the literature and also new ones, including TBX21 or CX3CL1. So this ranking seems promising, but we want to know, of course, if we can identify new enhancers that do regulate those genes. And for that, Elena cloned CRISPR activators and repressors. So here you can see dCas9 VPR mini, which is a pretty short form of a transcriptional activator that is linked to a Cas9 protein that is nuclease deficient. So it cannot cut, but by using a guide RNA, you can recruit it still to any region in the genome. And when we look at the PD1 locus and we recruit this activator to this 5 kb upstream enhancer of PD1, where we have a predicted peak to gene link here, we see that in comparison to control guide RNA, we see an increase in PD1 positive T cells here by simply targeting this enhancer. And we see this is significant. And we also see when we look at the PD1 MFI, the protein expression, the median protein expression measured by FACS is going up. We also tried the opposite. So we identified an enhancer and a loop to this enhancer at the TCF7 locus, which is active, supposedly active in the state of memory T cells, so the cells that also seem to play a role in anti-tumor response. And when we repress, when we target this region with the repressors, there's the SIM3 domain, which has been reported as a highly potent repressor. When we repress this enhancer, we see that from 5.88% of TCF7 negative cells, we get really a drop in TCF expression, and more than 1/3 of the cells lose TCF7 expression in our model. And this is significant percentage-wise, and also when we look at the protein expression levels by the MFI in FACS. And this region is 20,000 base pairs away. So I think the effect for me is still pretty astonishing how such a region so far away can control this gene in such a significant manner. Okay, so the very last part is just one more slide. I would like to show you how we intend to use this information to get a handle on improving T cells in a therapeutic setting. And this is a very nice collaboration together with Hendrik Böck's lab, Markus Perl, an MD, PhD, and Leonard Schmidl, a soon-to-be medical PhD, they have been picking from our ranking that is based on the cell state-specific open chromatin profiles and the connection to the enhancers. They've been picking additional genes that are not on this list here, but are still very highly ranked and understudied in the context of T-cell dysfunction, and try now to knock out these genes to make CAR T-cells better. So their approach is to isolate T-cells from PBMCs, to transduce them with a CAR, with a chimeric antigen receptor that can recognize specifically tumor antigens like CD19 or CA. Then they CRISPR out the candidate gene, and then they co-culture the CRISPR CAR T-cell with a CD19 positive or CA positive tumor cell to see if the knockout of the dysfunction related gene is improving the function of the CAR T-cell. So when we look at the knockout levels, because this is not so easy, many people do this now successfully, but still not so trivial. The knockout rates of the candidate genes is really nice. So we get 80 to 90%, sometimes even more, knockout of our candidate genes. So we picked 10 of them. And you see that the killing, so the specific lysis in percent here versus the control, is increased and decreased in some of the cases when you knock out a candidate gene. You might argue that the difference is not so big, so it's a slight increase, but you have to be aware that the CD19 CAR is a highly efficient CAR, and CD19 on the tumor cells is expressed at a very strong level. So this is already a, let's say, an extreme efficient model to kill tumor cells when you put together a standard CD19 CAR with a CD19 positive tumor cell. So a slight increase even here is already a remarkable observation. And when we look at the time-lapse killing of CD19 T cells of 3 of the candidates, you see that in comparison to the untouched T cells, so that did not go through the CRISPR procedure, 3 of the candidates, they show a faster and a better tumor cells type-specific lysis of the tumor cells. When we go towards a setting where the tumor antigen is not prevalent such a high density on the tumor cells, where the T cells get not activated so strongly via the CAR, when you look at CA target cells, now at the confluences, so now here less is better, so means more killing. You see that when you grow the tumor cells only, you see that they can grow nicely. If you add in green the CARs, the CA CAR that did not undergo CRISPR, you see already some blockage of the cell proliferation, of the tumor cell proliferation. But when we add our candidate 5, which is our favorite gene to knock out, we basically completely abrogate tumor cell growth and even go into a lysis of the cells to some degree in the end after some days. So to sum up the second part briefly, I hope I could convince you that the integrated single cell profiling can predict enhancer promoter interactions, also in human T cell states. That makes sense. We can validate some of those interactions with CRISPR IA targeting. And I hope you can also appreciate that this CRISPR IA targeting might enable us to fine-tune gene expression, because sometimes, especially in the T cell universe, it might not be good to completely knock out a gene, but to adjust the gene dosage. And the CRISPR IA, I think, by targeting enhancers, could be a good way to do this. And finally, in the last slide, I wanted to convince you that this ranking and comprehensive information about gene regulation, gene prioritization, and chromatin changes in the T cells helps us to select potential targets that we can modulate to finally improve adaptive T cell therapy, as we've been trying now in the CAR T cell setting. So this most of this data we just put together to a manuscript and uploaded on bioarchives. So I'd be very happy if you think the work is interesting, if you could share it. And I would like to thank finally everybody involved, especially the people in my lab that did this work. So this was Daniel Riegler and Elena Fernandez that have contributed most of the work. Of course, I would like to say thank you to the collaborating labs with the work on the CAR T cells from Hendrik Abken, Hendrik Pöck, and especially Markus Perl and Leonard Schmidl, and of course the NGS and FOX core units that help with the cell sorting and the sequencing, the DKFZ group from Benedikt Bross with Malte Simon and Charles Imbusch that contributed to the bioinformatic analysis, and especially also the university clinic collaborators. Without them and without providing the samples and being interested in the work, supporting us, this would not have been possible. So big thank you to everyone, and a big thank you to you for staying and listening, and I'm happy to take any questions. Thank you very much.
Wonderful. Thanks so much, Christian. That was a fantastic talk, really. It's a beautiful story that you just presented, from, generating 26,000 cells on a plate to describing the different epigenetic programs and actually knocking out genes for refunctionalizing exhausted T cells. So that's very fantastic already. And we really enjoyed the talk today. Thank you.
Thank you.
So I'm very happy, or we're very happy to take any questions that might be there. So I really encourage you to please raise your hand if you have one, or also feel free to write it directly into the chat and I can read it out for you. Actually, I see one popped up now from Camille, and it says, thanks for the great talk. You presented a trajectory analysis of an integrate data set in which you tried to remove the effect of the tissue of origin. How did you account for the tissue of origin in the trajectory analysis? Was this analysis based on splicing kinetics? Great question.
We used Harmony to correct for the tissue. So the Harmony package, I think, is pretty great. What we also saw that at some point, one principal component was basically containing all the tissue information, and that was filtered by the Archer package because it was correlating also to the sequencing depth to a very high degree. So this was a combination then of several factors that led to this kind of unifying UMAP where we built the trajectory on.
Great, thanks. I also have a question from Raphael in the message here. Raphael Aguelo. He was asking, in the t-SNE of CD8 T cells, it seems that 50% of PBMCs, the CD8 T cells, are mate. Have you confirmed this by other methods? What other cells express CBTB16 and CD127, etc.?
So this cluster 7 containing PBMCs is actually so the percentage of this cluster, I think you have to be careful because it's percentage-wise we sort on the plate from every sample a certain amount of cells. So this is not representative for their actually containment in the blood. But you are right. So these are, I'm pretty sure these are mate cells. We did not confirm, especially with those samples because they are scarce, the expression of ZBTB16, our PLFZ, in those cells by flow cytometry. But we have another project where we profile mate cells, and we see we have some donors where we have actually more than 10% of mate cells in the peripheral blood. And as apart from the PBMCs, the liver, when we look at the other half of the of this cluster 7, so one half the PBMCs, the other half is dominated by the liver cancer, and there it's known that there are many mate cells. But from the PBMC origin, it's true, it's quite some of them, but we see this really in a donor-dependent way that many of the cells in peripheral blood, they could be classified as mate, but it's varying a lot. So it's from, I don't know, 1-2% to sometimes 10%, depending on the donor. Did this hopefully answer your question?
I hope so. I think so. It sounded good. Unfortunately, Raphael let me know that he can't stay for it. So hopefully he'll listen to the recording to get the response. And then I think we have time for just one more question. So I see Barbara Hulbacher has her hand raised.
Hey, yeah, so thank you very much for the cool talk. So I might have missed this, but so you presented that you integrated the ATAC-seq data and then you use it to predict enhancer gene interactions. Can you maybe explain a little bit how that works? Because we all know that actually assigning an enhancer to a gene is actually kind of tricky, especially with like 3D chromatin structures and so on. So do you know how that works in detail?
So this Archer package, I think uses some of the Seurat functions to do this. So it basically aligns the chromatin accessibility-based gene inference of gene activity. So we have, based on the chromatin, we have the inferred gene activity, which is built on a model that has been validated and tested. So you can predict basically gene activity from the chromatin. And then you have the RNA expression on the other side. And Archer is putting this together. So it looks for every cell in the ATAC-seq based on the genome-wide chromatin activity. It picks a cell from the single cell RNA-seq, as far as I understood it, that is matching most closely in terms of gene expression. And then it builds pseudo single cells, multi-omic cells based on these two matrices. And then in this new multi-omic single cell data set, it basically looks for correlations of if a region is open and which genes in the surrounding are then expressed, or if the region is closed, and again checks the surrounding genes if they are expressed or not. And if you do this over many single cells, you can build correlations of the accessibility of a peak and of the gene expression of the surrounding genes in a certain distance. So this is not genome wide; it's limited to you can put it in the settings how far it could look. And I think it's calculated now for maybe 100,000 base pairs or more. And there it builds these correlations between accessibility and gene expression in these pseudo multi-omic cells. When I got this right, I think that's the way it's done.
Thank you very much.
Great. So we're a little over the hour, but there was one question that came in. So we'll quickly answer it maybe from Denise Duma. When you find multiple cell-specific enhancers, do they tend to regulate the same set of genes, or is each gene regulated by different enhancers?
So as we have filtered for cluster-specific peaks, we do not cover, let's say, overlapping enhancer sets at the moment. But I think this is a good idea. People have shown that this can happen, that enhancers jump or they change their targets or that they co-regulate. But we were really focusing on, let's say, the most specific way that we could be, and therefore we filtered really on only the cluster-specific peaks. So I'm actually not sure if one enhancer that is specific for terminally exhausted T cells is also regulating the precursor exhausted T cells, which could be the case if the peak is also open to some extent. But I can't say. I think that needed to be tested in a complicated setting. But that's a good point. Thank you.
Okay, great. Well, I think we'll have to end now. We're a little over time. So thanks again so much, Christian, for this lovely talk. You really presented a great story today and initiated lots of interesting discussions. So we all thank you for presenting. And I will remind everyone once again that the next lecture is on May 20th. So we hope to see you again then. So thank you very much for joining us today.
Thank you.